Hi everyone, I'm Murali Krishna, working as Associate Professor in Engineering College of Pharmacy. Today I want to discuss about enzymes. So while we are discussing before going to the enzymes, so what is the syllabus for the enzymes in the biochemistry? So in the syllabus of biochemistry for B.Pharmacy 1.2, introduction, properties, nomenclature, IUB classification of enzymes, enzyme kinetics, Michaelis Menten plot, Lineweaver product plot, enzyme inhibitors with examples, regulation of enzymes, enzyme induction and repression, allostetic enzymes regulation, therapeutic and diagnostic applications of enzymes and isoenzymes, coenzyme structure and biochemical functions. These are the syllabus in the first year from biochemistry. So for every person requires energy. So nutrients are the one of the source of energy as well as being molecules, organisms required to grow, reproduce or repair. So generally we will require all these nutrients for the whatever the cells to grow, reproduce and repair. Biochemical reactions are the reactions or process used for the formation, breakdown and rearrangements of molecules to provide organisms with energy. So these are the whatever the chemical reactions used for the formation or breakdown or rearrangement of molecules to provide the organisms with energy. Those reactions we will call as biochemical reactions. Activation energy is nothing but how can you define the activation energy means the activation energy is the required input of energy to make a reaction start. If you see the graph between free energy and reaction coordinates, whenever a reaction starts with the reactants, generally we know the reactants will be converted into products. For those the reactants to convert the reactants into products, generally the energy requires. For example, in the diagram or in the slide, they are pushing the car. So when the car they are pushing means so for the car to start something, to start reaction, generally energy requires. So the energy required, whatever the amount of energy to make a reaction start is nothing but activation energy. So catalyst means a catalyst is a chemical which speeds up the reaction but is not used up in the reaction. Catalyst is generally a chemical which speeds up the reaction but is not used up in the reaction and which lowers the activation energy need to start a reaction. It is not used up during the reaction. It is unchanged after a reaction. Generally, the lecturers working at para coaches, so those are always ready to give some coaching time. They always do their work. They are ready. When they are giving some coaching means the students are getting. They are going to helpful in when their working time is going to be useful. So at that time means how can you correlate with this thing means because they are going to speed up the reaction but they are also changed. They always do their work. They are not going to change. They motivate the students like that. A catalyst means is a chemical which speeds up the reaction but is not used up the reaction. Lowers the activation energy need to start a reaction which is not used up during the reaction and it is unchanged after a reaction. So enzymes will also act as a biocatalyst. Enzymes are the proteins which speed up the rate of the reaction. They found in the cells throughout the body and lowers activation energy. So here the concept is enzyme we will call it as a biocatalyst, which is nature of protein which speeds up the reaction found in cells throughout the body which lowers the activation energy. Increasing the temperature make molecules move faster. Biological systems are very sensitive to temperature changes. Enzymes can increase the rate of reactions without increasing the temperature. They do this by lowering the activation energy. How can they are going to increase the rate of the reaction without increasing the temperature? Means they are going to decrease the activation energy. They create a new reaction pathway, a shortcut. For example, if you see here in the graph, if you are going to take the time between the graph between time and relative amount of energy in a molecule, so you can observe the substrate is going to convert into a product without the enzyme, and this is the with the enzyme. So without the enzyme, how much time it is going to take? With the enzyme, it make a reaction start shortcut. So they are going to reaction shortcut. This is a new shortcut. They make new pathway. Enzyme controlled reactions proceed 108 to 1011 times faster than corresponding non-enzymatic reactions. So the substrate of an enzyme are the reactants that are activated by the enzyme. Enzymes are specific to their substrate. The specificity is determined by the active site. So generally enzymes are very specific to their substrates. The specificity is determined by the active sites. Enzymes are going to helpful to convert the reactants into the products. What is the active site? One part of an enzyme, the active site is very particularly important. The shape and chemical environment inside the active site permits a chemical reaction to proceed more easily. For example, in a classroom, the environment is helpful to make a, the students are very helpful for the studying purpose. So in our, but when they are going to, when they are homes, it will not be accurate. So the shape and chemical environment inside the active site. Here in the college, the office, or whatever the classroom, they are making that environment to study. They are going to helpful making the study. Like that, the shape and the chemical environment inside the active site permits a chemical reaction to proceed more easily. What are the cofactors? An additional non-protein molecule that is needed by some enzymes to help the reaction. Tightly bound cofactors are called prosthetic groups. What do you mean by the cofactor? Cofactor can be defined as a which is a non-protein molecule which is needed by some enzymes to help the reaction. Tightly bound cofactors are called prosthetic groups. Cofactors that are bound and released easily are called coenzymes. What do you mean by the coenzymes? Means cofactors which are bound and released easily are called coenzymes. Many vitamins are coenzymes. Here we can, in the multiple choice, we can get the, like all cofactors are coenzymes, but all coenzymes are not cofactors. This is the true. So multiple choice, and when they are going to write some examinations, we have to read thoroughly. And what is the concept here? Many vitamins are coenzymes. What do you mean by the cofactor? Means any additional non-protein molecule is needed by some enzymes to help the reaction. Tightly bound cofactors are called prosthetic groups. Cofactors which are bound and released easily called coenzymes. Many vitamins are coenzymes. So properties, specificity. What are the enzyme properties? Means absolute specificity. The characteristic that an enzyme acts only one substrate. For example, glucokinase, which is also called as hexokinase, which phosphorylates only glucose. Urea is the only substrate for urease. Relative specific. The characteristic that an enzyme acts on several structurally related substrates. Hexokinase can catalyze phosphorylation of glucose, galactose, and mannose. So there are two types of specificities are there, absolute specificity and relative specificity. The characteristic that an enzyme acts on several structurally related substrates. Absolute means only one substrate. Relative means they are going to act on several structurally related substrates. Hexokinase can catalyze phosphorylation of glucose, galactose, mannose. Galactose, mannose are the epimers of glucose. In the isomerization, we know that galactose and mannose are the epimers of glucose. They are structurally related. They are made up of 6 carbons only. So hexokinase can catalyze phosphorylation of glucose, galactose, and mannose. Specificity. Stereochemical specificity is an enzyme's ability to distinguish between stereoisomers, L-amino acid oxidase. Oxidase is only L-amino acid into alpha keto acid and ammonia. D-amino acid oxidase. Oxidase is only D-amino acids into alpha keto acid and ammonia. An enzyme's ability to distinguish between stereoisomers. Stereochemical specificity means an enzyme's ability to distinguish between the stereoisomers. The process of unfolding is called denaturation. One more property is nothing but denaturation. So generally proteins are made up of amino acids. So those they are going to have proteins are existing in the primary, secondary, tertiary structures. So when the tertiary structures it will be in a folded protein. So when they are heating, here example you are seeing that egg when you are eating, the albumin is going to be breakdown. So the whatever the amino acid chains they are going to primary, they are going to quaternary structure. It is going to break into primary structures. So unfolding is going to be happen. So folded protein, well, when you are going to heat, then it is going to convert into a unfolded protein. That is nothing but one of the concept of what is the property, specific property. That is nothing but denaturation. Denaturation is nothing but it's a specific property of a protein, which is nothing but here we can say that is that one of the property of enzymes, because enzymes are nothing but proteins. Enzymes are made up of proteins. So nomenclature, most named for substrates for reactions. So generally, when you see the nomenclature, generally you know the so many types of enzymes in our body. So for those we have classified the name of an enzyme in many cases end in ase. For example, sucrase catalyzes the hydrolysis of sucrose. The name describes the function of the enzyme. And oxidases catalyze the oxidation reaction. Sometimes some common names are used, particularly for the digestion enzymes such as pepsin and trypsin. Some names describe both substrate and the function. For example, alcohol dehydrogenase oxidases ethanol. Enzymes are classified into 6 functional classes. Mainly, by the International Union of Biochemistry. Who introduced this system is IUB, International Union of Biochemistry, classified the enzymes into 6 classes: EC1, EC2, EC3, EC4, EC5, EC6. EC1 means oxidoreductases, EC2 transferases, EC3 hydrolases, EC4 liases, EC5 isomerases, EC6 ligases. So here principles of the international classification means there are where basically, for example, if you see EC 2.7.1.1 means hexokinase. What do you mean by 2 here? 2 is the class, which is the transferase group. A7 is the subclass, transfer of phosphate group. 1 is the subclass, alcohol is the phosphate acceptor. So this is 2.7.1.1, and last one is the specific name, hexokinase. So total 2.7.1 represents hexokinase. Hexokinase catalyzes glucose to glucose 6-phosphate. Glucose plus ATP gives glucose 6-phosphate. So total classification: oxidoreductases, transferases, hydrolases, liases, isomerases, ligases. In the slides you can see the examples. So first class, oxidoreductases. The enzymes are going to helpful in making a reaction, oxidation and reduction. Example, lactate dehydrogenase, which involves in the oxidation reduction. Transferase, group transfer. Generally, whatever the phosphate group transfer, so many enzymes they are going to helpful in transfer of amino groups. Amino transferase can help in amino transfer of amino group. Hydrolases, hydrolysis reaction, transfer of functional groups to water. Primary liases, additional or removal of groups to form double bonds. Example, fumarase. Isomerases and isomerization, intramolecular group transfer, that is nothing but from aldehyde to ketone, ketone to aldehyde. This is nothing but isomerization. Intramolecular group transfer. Ligases means bonding. They are making two substrates getting together, linking together. For example, amino acid, tRNA, those things will helpful in the ligase, making the bond, making the two molecules are bonding together with the hydrogen bonds or anything. So the mechanism of action of this enzymes, Michaelis and Menten concept. When the enzyme